you will be able to tell me what the pointer is pointing at. What is that little structure right here? You know the big cough drop? Okay.
Ciliary body.
Perhaps, yeah, part of the ciliary body. If you said, well, it's part of the ciliary process, maybe. Usually there are also little tiny little ligaments you can see, kind of, they look hairy, kind of, they're like connected. Those are the suspensory ligaments. So you're looking at a piece of the ciliary body underneath the lens in this case. What is this structure right here? Or if I made it real clear, actually, right up there. That tunic, superficial tunic, most superficial tunic of the eyeball. Cornea, probably cornea out here, because here's the lens, so in front of all this is cornea. This is probably sclera, right, the white part of the eye, and superficial tunic or layer of the eye. Okay, cool, let's, I don't know why I keep getting big screen. There we go, there it is. What's that structure? Lens. Now you can see them a little better here. See these little hairy, they almost look like little pieces like this, attaching? Those are the suspensory ligaments, connected to the ciliary process of the ciliary body. This whole part is the muscle of the ciliary body. What's this structure right here? I'll just step back for a minute. That. Caroline? Iris. Yep, iris. And if we move up a little further, there's an opening or a gap right here. That's the pupil. Okay? Keep going. What's this one? Now we're in the anterior segment of the eye. Now we go back to the posterior pole of the eyeball. What is that? Retina. Yep, this is the retina. Those are all those cool little layers of the rods and cones, right? Bipolar cells, ganglion cell axons, and then this is the pigmented layer of the neural layer, and then the choroid coat. So the vascular, capillaries, melanocytes, all that stuff, right in the back of the eye. Okay. Photoreceptors, as I said, there's two types. There's the rods, they're more sensitive to light, so I call them the black and white. Those are like your black and white photoreceptors. They allow vision in dim light. These are functioning when you like walk into a dark room, when right dawn and dusk, or real low light candlelight, low light conditions, your rods are functioning. Cones operates best in bright light. So this allows us to have high acuity or good clarity of vision, color vision. So cones, color vision, CC. That's how I remember. They are neurons. Whenever you hear photoreceptors, hair cell receptors, audition receptors, all the special senses, the receptors that allow your special senses to be taken in, interpreting energy and putting it into action potentials, those are all neurons. They're all specialized neurons. They're crazy looking, but they're neurons. Question?
Yeah, are we the only mammal that doesn't see good at night or in the dark?
No, there's lots of other mammals that are not nocturnal. Like all the other great apes, pretty much like us, like orangutans and gorillas and chimpanzees and stuff. They go up and climb up in their trees and go night-night and make little hammocks, and they don't see good at night. They're not nocturnal. Mammals are split, kind of diurnal means they function during the day and they do all their business during the day and they sleep at night. Nocturnal, it's reverse, they're up at night. It depends on where they live, what they eat, their behavior, what they prey on, all those different things feed into that. Dogs are pretty much more diurnal like we are, although they tend to be more active in evening hours like coyotes. We have a lot of coyotes around here actually. So in the morning, I get pictures, I got video of coyotes over here, not even afraid of me either. It's very cool. But cats totally nocturnal. It depends. There's nocturnal and diurnal. But they have differences in their rods and their cones, and also the back of their eye, their choroid, as you've seen. In the cow, you would think, well, they're not really nocturnal. They're out chewing during the whole day. But they are out at dawn and dusk hours, so when it's low light, that's when they're sort of the most active. Yeah?
I know like everybody's different, and sometimes eyesight with like blindness kind of depends on genetics, but is it true that like different color eyes have a different kind of clarity or vision, when it comes to night sometimes?
No, not that I know of. No, like light eyes, blue versus brown.
Yeah.
No, not as far as their accuracy, their acuity, their clarity, their low light, how their cones and their rods function. No. But we do have color blindness. People genetically that can be missing a certain photopigment, and so they can't make out differences, for example, between green and red. So we have red-green color blindness. It's actually carried on the X chromosome. So almost everybody affected with it are male. And every time I do physio, I'll have one guy in there at least that's colorblind that didn't even know he was colorblind. It's pretty interesting. Females never. Why? Why females almost never? Like 1 out of 100,000 or something crazy. Why?
Because
Two
we're
X.
So on the X chromosome, females have 2 X's. So if you have a messed up one on one X, you gotta back up with another one on the other X. So those X-linked traits, some of those like male pattern baldness, color blindness, I hate to say it, all those, right? Males have one Y, one X, so they get the jacked up gene on the X, that's it, you're gonna have it, there's no way getting around it. So genes code for eye color, they code for also visual acuity, so shape, that comes down to what you're asking to is shape of the lens, shape of the cornea, shape of the eyeball. There are genes that are linked with that, the anatomy of the eye. And so you can see whole families where they all have prescriptions, they all have to wear glasses, they all have contact, not just age related, but they get diagnosed early on with having visual acuity problems. That comes to structure of the eyeball. So basically the light comes in, I don't really go over this in anatomy, I go over it in physio, because we go in deep in physio to the eyeball and how it works. But basically the eyeball is either too short or too long. So those light rays get bent by cornea, fluid, lens, boom, go back to the retina, they either fall behind the retina, or they fall in front of the retina. So you have to make lenses that are either concave or convex to bend those light rays and get it back where it needs to be, that's it. So, genes coding for different things, but yeah, no, I don't know if color itself affects any of those things in general. I would say no, because a lot of it is coming down to the genes. All right, visual pigments though, that's how your rods and your cones work, is they have visual pigments that get cleaved by light, and then it activates a signaling pathway inside of those cells. So we have rhodopsin is the pigment inside your rods. Rhodopsin in your rods. Here's your rods, here's your cone, okay? 3 opsins, other color pigments, are in your cones, and those detect green wavelength light, red, and blue wavelength. Okay, so your cones are the ones that are detecting different wavelengths of light. And you guys learned about the visible light spectrum a little bit, if you ever have. It goes like the true rainbow. When you look out and you see refracting light and it makes a beautiful rainbow, right? Red through purple. Those are different lengths or wavelengths of light traveling through air, right? Some have a bigger sign, a frequency or a wavelength, and some have a shorter one. So those longer, intermediate, and shorter wavelengths are what your cones actually are able to respond to depending on which pigment they have. So if they have a certain pigment like the green pigment, they will send action potentials when they're exposed to green color. This is what allows you to see color vision. This is what makes us trichromats, all primates including us. Great apes down to the little New World monkeys like the macaques and everything. We're all pretty much trichromats. Chrome means color. Chrome means color. So we're trichromats. We have 3 sets of color pigments. So we can see all the rainbow and all those variations of green, red, and blue, and you're never seeing anything pure. Nothing is almost ever pure. It's always mixes of those colors, right? And how much of those wavelengths are striking the backs of your retina and telling your cones what you're seeing. But not all mammals, not all dogs, or animals like dogs, for example, cats, they're not all trichromats. They're like dichromats. So you probably heard, oh, your dog sees in black and white. That's not true. They don't see in black, but they see color. They just don't see the rainbow of colors that we see. Yes? Okay. What does that mean, trichromat? 3 photopigments, so you can pick up reds, blues, and greens. You can see the rainbow of colors that we see, which are combinations of those. These are primary colors, by the way, in the artist wheel, like many of you do art stuff, these are primary colors you can mix together and get all the other colors. All right. Light and heat can damage your photoreceptors, which are neurons, and they're very, very precious. You have to take good care of them, so that's why they're tucked and they're the furthest away from the pathway of light coming into the back of the eye. They cannot regenerate. We don't have stem cells for rods and cones. They're continuously replacing their outer segments. So actually the epithelial, the melanocyte, the pigmented layer that makes the melanin and keeps it dark and allows, prevents a lot of scattering of light and disrupting the rods and cones. We have that dark choroid layer in the back. It actually, those epithelial cells back there eat the outer segments of the rods and cones. They sort of digest them and break them, and then the rods and cones regenerate new outer segments. So they're always making new color and rhodopsin pigment and color pigments and beefing up that outer part, because that's what's getting struck by photons and activating those neurons so that they're telling you, oh, you saw red, oh, you saw bright light, oh, you're in a dark place, like giving you all that information. So if you look closely at the back, where exactly are the photoreceptors? So here's the pathway of light coming in, here's the pigmented layer, okay? And the vascular blood vessels and all that stuff. So where actually are the rods and cones? Are they here? Are they here? Are they there? last one, so they're closest into that pigmented layer right there, so they're tucked away. So when you look under the microscope, you'll be inclined to go, oh yeah, they're right here, because light is coming in, boom, hits right there. No, they would bake and fry and die, fall apart. They really have to be protected, so they're the furthest back away from the incoming path of light. Okay, so that's, regional specializations. There's this neural layer where it ends at the back of the ciliary body, and that's where the neural layer begins, the retina, and we call it the ora serrata, like an aura around your eyes. It's around the ciliary body. That's where the retinal layer begins. Pigmented layer goes all the way up over the ciliary body though, so you can see it's brown. This all looks brown on the back surface because it's got all the melanocytes, the melanin in there. Macula lutea, this is an area in the back of the eyeball. I think I drew a bullseye here. Yes, I did. Macula lutea is like if you have this bullseye in the back of the eye, a little off center from the optic disc. We call it the macula lutea. It's actually on our eyeball model, and if you were to open it and take it all open, you look and there's this little pink sort of spot. Macula lutea is mostly cones. The fovea centralis is like the dead eye bullseye right in the middle of that. This is tissue in the back of the retina where this is all cones and this is mostly cones. There's also some rods here, but in the dead center, the fovea centralis, that's all cones. That's the region of highest visual acuity. That is why when you look at something, if you really want to absorb the detail and see with clarity and have good visual acuity, where do your eyes have to be? Where do you have to focus? Like say you're looking up here, where do your eyes, where do you have to get that image to come into your eyes?
The middle or like the center?
Right in the center, dead center, right? And this comes when someone's talking and you're paying attention to them, right? You're really focusing on their eyes, their mouth, like you're looking at that straight in the center of your eyes, because if you notice that in your periphery right here, you don't have good acuity. It's all blurry. We can see movement. There is a true thing too, of course, seeing that out of the corner of your eye. You guys have heard that, oh, I saw it out of the corner of my eye. Came from, you can see movement because your rods are mostly on the outer part of your retina. Cones are mostly in the middle because they're your high acuity color vision. So your rods are detecting low light and movement. So if something goes by, like you feel like, oh, there was something there, just some little, that's your rods working and going, yep, there was movement we detected. You can't make out good clarity or good acuity, but they'll pick up. So this area, all cones, the area around it, the macula, mostly cones. Highest visual acuity, so you really have to get that dead center coming into your eye. So that's also why you do those visual acuity tests, you know, where you do the E's. We're gonna do that in physio, actually, the different letters, you know, you cover your eyes and you do them. You have to look just kind of straight at it, and you can't look at it sideways or you have to directly look. All right, optic disc, you should know where that is. What's the optic disc? What does it correspond to when we talk about the function of the optic disc? Yes? Y'all know what that is? Okay. I have this picture of this eyeball 8 million times in here. Okay, so you'll know. But macula lutea, just to show you though, isolate these structures I put in blue. Macula lutea, fovea centralis right in the center. Highest concentration of cones. Millions of cones, all just right in that little tiny spot. Most of the rest of the retina is rods, and a little rods even here, but fovea centralis is the only cones in that location. Optic disc again is right there. Oops, you have a blood supply in the back of the eye, so here's the retina. Optic disc, that's just the ganglion axons going back and giving rise to the optic nerve. You've got a lot of central arteries and veins, and then capillaries that branch off from those. We can view this by light, right? Ophthalmist, so you can go right in and just view the back of your eyeball, look at your retina, look at the blood supply of the retina, make sure everything's fine. You've got different fluids in here. You've got the anterior segment that has, these contain aqueous humor, so all of this. And then you've got the anterior chamber that's in front of the lens, the posterior chamber that's surrounding the lens. All of these have good nutrient aqueous humor fluid that gets circulated and then taken into the venous sinus, goes back to circulation, and then new fluid is actually created and made. So it's constantly circulating inside the lens in the eyeball. The lens, transparent, thick, biconvex, means like cough drop, right, so it's like that. Held in place by the suspensory ligaments. The epithelium, it covers at the surface, and then it's got lens fibers, so these actual connective tissue sort of fibers that make the bulk. They're made continuously throughout life, so they're added. And your lens actually kind of enlarges throughout life. So you start with a certain size lens when you're a baby, and those lens fibers and epithelial cells kind of keep getting made throughout life, so they get bigger and bigger. When you go from, you know, baby to adolescent to adult. The eye obviously is an optical device. It's basically how we've made cameras and microscopes and all these things based on the human body and how the eye is this amazing, light imaging viewer. So structures bend. In the eye, they bend light in different ways, different types of bending. Light rays end up converging on the retina at a single focal point, okay? And what happens is if you have, here's accommodation for a near target, lens is bulging up, here's accommodation for a target that's far away. Makes it very, very thin. I'm not gonna belabor this too much, but I want you to know what accommodation is. In physio, you're gonna have to learn all about farsightedness, nearsightedness, visual acuity problems, astigmatism, accommodation, but I want you to know about accommodation in here. All right, light bending structures, what are they? The refractory media or structures of the eye that work as this amazing optical device, like a microscope, are the lens, the cornea, and the humors, the fluids. That light is bending a certain degree every time it goes through one of those structures. So they work together with the main roll coming down to the lens to bend light and get it back on the back of the retina. Can someone tell me what time it is? I wanna make sure I don't go, I have no.
12:36.
Okay, cool. All right, accommodation, as you said already, Ty, basically is accommodating to look at the eye can function, looking at something close or looking at something far. That's it. As you get older, less able to do that because the elasticity of the lens, the pliability of the lens goes down. Visual fields and pathways to the brain, we have several different things going on, but I want you to remember, primary visual cortex is in the back of the occipital lobe. Those are the neurons that this is processing what you see, what colors you see, what light intensity you see, the information that you see. This is where it's first being processed. I mean, major processing of vision. But notice you have, here's a fixation point, both eyes. There's overlap between the left and the right visual fields and the left and right eye. And there's crossing over those fibers. So the outer retinal fibers, the ganglion cells, go back and they stay on that side of the brain. But in the inner half of that eyeball, it's contralateral and it crosses over and goes to the other side of the brain. Essentially color coding this visual field, just to keep it straight, the right visual field in left is processed by the left side of the brain. The right side of the visual field crosses in both eyes, it's hitting over here on this one, it's hitting in the medial aspect in this one. The right half of the visual field crosses over and is processed by the left side of the brain. So there's this contralateral processing and crossing over. You've already seen this in lab, you've already seen these in lab, you've dissected the entire brain in lab, right? So kind of putting all these together, optic nerve, what's the crisscross? Optic chiasma, optic tracts, and then there are midbrain structures, okay? They have contralateral crossing, so these go across here, and these go across over to that side. Let's see, what other structures I was gonna point out? The thalamus, so here's the thalamus, and then also, remember that in the midbrain, the inferior and superior colliculi? Remember the midbrain in the back of the sheep, you can even see them, those protrusions that stick out of the back? These synapses in the superior and inferior colliculi, and then in the thalamus, the sensory relay station, and then synapse on another set of neurons that go back to the primary visual cortex. So now the brain gets to process, okay, what you're actually looking at. What's the color, what is it, what's the form, where is it, all that. And then actually talks to lots of other parts of your brain and says, okay, what exactly am I looking at to make sense of it? Okay, a lot of disorders of the eye and vision. Age-related macular degeneration, this is one. This happens sometimes in older individuals where the retina and the macula, lutea, they just detach, they come right off the back, okay? So the macula begins to generate or lift off the retina. It involves the buildup of visual pigments in the retina, leads to anywhere from blurred vision to blindness. We also have retinopathy of prematurity. Retinopathy is this pathological situation of the retina that happens with premature babies, okay? So blood vessels grow in the eyes of premature infants, and sometimes what happens is these have blood vessels are new and not mature, and they have weak vessels, and they explode, and they cause hemorrhaging, loss of blood in the capillaries in the back of the retina, and blindness, because those rods and cones and all those tissues don't get oxygen, they don't get nutrients, and they die. This is why some babies that are very premature will go blind. So blindness is a risk for being premature, because it's retinopathy of prematurity. Also, we have various types of blindness, congenital or acquired, many different causes, genetic, viruses, infections. Long time ago when they didn't really do vaccinations right before modern medicine, some of those viral infections would actually get into the head and the brain and the eye, and they would cause blindness. So you could go blind by some basic childhood diseases, or congenitally be born with it. So either genetic or lacking a part of the structures, the visual pathways were actually messed up from the very beginning. Okay, I'm going to come back to the ear, I'm gonna skip the ear now, and I'm gonna go to the overall of the heart, so the cardiovascular lecture slides. Because you guys are gonna be dissecting a heart, so I wanna spend about 10 minutes going over the overall structure, so that you know what you're looking at when you get in lab, and that everybody's on the same page. Okay, so I'll come back to the ear. We looked at the cochlea under the microscope, we looked at the ear model, but we're gonna now dissect the heart. So what I'll talk about is the pulmonary and systemic circuits. This is cardiovascular and circulatory system, are all one, kind of heart, blood, and blood vessels. Heart structure itself, okay? Current research, a little bit of that. Conduction system, so how the heart is able to pump. Cardiac cycle, how it's able to pump blood, okay? And then some clinical implications and disease and outcomes of the heart. Then we'll go into blood, then we'll go into blood vessels. So this is all cardiovascular stuff. Disorders and diseases at the end, and the effects of aging and exercise, just like always. So we're gonna be on this for a little bit. So we'll talk about, we'll get through heart, all the way through heart on Wednesday. So on the lecture exam, goes through heart. But then the rest of cardiovascular will be on the next, will be on your final. So the blood vessels and all that other stuff, okay? So cardiovascular, AKA circulatory system. I just have this little thing. It's an amazing piece of machinery. It's a vital organ. It keeps blood pumping. It keeps all of your tissues oxygenated and functioning well, including itself. And this is what happens in a heart attack. The heart gets, its own blood supply gets blocked and damaged, and so it can't get blood to itself, and that tissue dies. And if your heart dies, that's your pump, you die, unless you can bring it back, right, and get it functioning again. I'm gonna skip this for now. I'm gonna skip this and just go straight to what it does. What is the heart? What does it look like? It's a pump. Basically, it's a double pump. When you dissect your hearts in lab, you'll see there's little chambers on the top and big chambers on the bottom. You got your atria and your ventricles. It's a double pump. And then it's got a couple different circuits. You have a pulmonary circuit and a systemic circuit. You have the circuit of blood vessels that leave the heart and go to the lungs. The lungs are where you get oxygen, right? And you gases exchange, so you get CO2 going out and then O2 coming in. So that whole circuit is the pulmonary circuit that comes back to the heart now with oxygenated blood. And then we have the systemic circuit. This is your system, your whole body. This goes from your heart, pumps that oxygenated blood to your head and your body. And then it returns once your tissues have taken up the oxygen, released their waste, all that comes back to the heart. So that's the systemic. Vessels transport blood to and from the body tissues. We have the atria, or the upper chambers, the ventricles are the lower chambers. The main muscular ones, and you'll be surprised in lab when you see this, the atria are very, very thin and small, and they're on top, and they're super easy to cut through. They're very thin. We've got auricles, or little ear flaps sticking out on top of them. They look strange, but they're not the big muscular pumping action of the heart. That comes down to the ventricles. The whole bulk of the heart is the ventricles, and that's where those are the ones that really pump. Pumping out to the lungs and pumping out to the body. And we'll go over the heart in labs and stuff too in detail, like what are the different parts and how do they look. Pulmonary circuit is here, systemic circuit is here, okay, representing your whole body and your head, representing your lungs. So here's the heart, and there's these two complete circuits, and the reason we call them a circuit is because literally they make a full complete circle, a circuit that starts at one point and ends at one point. So the pulmonary goes from the heart, and actually anatomical position, so what side of the heart is that?
Right side.
Yes, perfect, you have to remember to switch it. When you guys are dissecting, you're gonna have to do the same thing, right? It's not how it faces, it's how it faces you in anatomical position. So it's like a person in front of you looking straight at you. That's the right and that's the left. It's the opposite of your right and left. So it leaves from the right side of the heart, goes to the lungs, picks up oxygen, comes back to the heart, to the left side of the heart, and then from the left side of the heart, through the aorta, this is actually the biggest blood vessel in the entire body, the aorta. You'll be able to see it now. It looks like a stem sticking straight out the top. This aorta goes out to the systemic circuit, serving the neck, the head, the whole body. And then in the capillary systems, get all the diffusion of gases and waste, everything happens, and then the deoxygenated blood goes back up and dumps into the right side of the heart, closing that circuit. So here's systemic, here's pulmonary circuit. We're gonna go over these different parts. What are these two chambers that are on the top right here? Here and here?
Atria.
Atria. So these big muscular ones down here are what? Ventricles. Now, I'll tell you, looking at these, when you dissect these, it's hard to tell. They look kind of even in their muscular wall, and it looks like steak. So if you're hungry during lab, you end up feeling like, well, this kind of looks like steak. It's gross. You're like, no, that's not gonna happen. But I'm always hungry in lab, so all of it ends up looking like food to me, okay? But it looks like steak, and you'll see that the left side of the heart, okay, so here's the left, here's the right. The left side, the one that has to pump out to this massive resistance of all the capillary beds of your entire body, is way more muscular. So thick, if you dissect the card, and I always have to just ask, which one's the right, which one's the left? Just look for the thickness of the steak, okay? The thick steak, left side. You don't even have to know any other vessels, just look at it and go, wow, that's super thick. Right, it's just like this thinner little side that's collapsed down into this little crescent. Well, that's the right side. It's just pumping out to the lungs. The lungs don't have as much resistance or resistance to blood flow and capillaries as the rest of the body has. All right, that's kind of gross, sorry. But that's how I can look. I can cut in any section and go, oh, that's left. There's right. I can figure it out right away. Heart typically weighs about 300 grams, healthy heart. Okay, so couple pounds, you can hold it in your hand. We're dissecting sheep hearts. They're very similar in size to adult hearts. And actually, you guys have probably heard that, well, if you hold your hand, that's the size of your heart. It's really not so accurate, that's a myth. It's more like 2 of your hands. It's more like that, sort of. That's about the size of your heart, okay? So our sheep hearts are a little smaller than that. They're about there. Largest organ of the mediastinum, remember that? Located between the lungs, the compartment. Yeah, it's about that size. It's a little smaller than that. Closer, and it sort of has from childhood to adolescence to adult, it changes a little bit. Located between the lungs, there's the apex and the base of the heart, okay? So where it sits. So here's the thoracic cavity, here's the diaphragm. It is literally sitting on top of the diaphragm. Here's the heart itself. It's got apex, it's got a point, and then it's got a base that it sits on. So the heart is actually like this. It's actually in the center of the chest, behind the sternum, protected, and it kind of sits back and tilts to the left. So that's where the point of the heart actually doesn't go straight down, it kind of goes like that, and it sits back, okay? And it's sitting on top of the diaphragm, located in the mediastinum, between the 2 lungs. And actually your left lung has a cardiac notch to make room for the heart, because it has that